Continuing with the subject on section H4, microRNA, we deal with the genetics of microRNAs, miRNA, which are single-stranded RNA molecules of about 21 to 23 nucleotides in length, which regulate gene expressions. miRNAs are coded by genes that are transcribed from DNA, but not translated into protein non-coding RNA. Instead, mature miRNA molecules are partially complementary to one or more messenger RNA, mRNA molecules, and their main function is to down-regulate gene expression. They were first described in 1993 by Lee and colleagues in the Victor Ambrose lab. Yet the term microRNA was only introduced in 2001 in a set of 3 articles in Science, 26 October 2001. As of early 2000, computational analysis by IBM suggested the existence of as many as 50,000 different miRNAs, and the typical mammalian cell, each with perhaps 1,000 or more potential target of the nucleus. 1 second. Formation and processing. The genes encoding miRNAs are much longer than the processed immature miRNA molecule. miRNAs are first transcribed as primary transcripts of primary miRNA with the cap and poly A tail, processed as short 70 nucleotide stem loop structures known as pre-miRNA in the cell nucleus. This processing is performed in animals by a protein complex known as the microprocessor complex, consisting of the nuclear trochanter and double-stranded RNA binding protein pasha. These pre-miRNAs are then processed to mature miRNAs in the cytoplasm by interaction with the endonuclear dicer, which also initiates the formation of RNA-induced silencing complex RISC. This complex is responsible for the gene silencing observed due to miRNA expression and RNA interference. The pathway in plants varies slightly due to the lack of trochanter homologs. Indeed, dicer homologs alone affect several processing steps. The pathway is also different for mRNAs derived from intronic stem loops. These are processed by a dicer but not by trochanter. Either the sense strand or antisense strand of DNA can function as templates to give rise to miRNA. Efficient processing of pre-miRNA by trochanter requires the presence of extended single-stranded RNA on both 3' and 5' ends of happen molecules. These ssRNA motifs could be of different composition while their length is of high importance. If processing is to take place at all, a bioinformatics analysis of human and fly pri-mRNAs and a bioinformatics analysis of human and fly pri-mRNAs reveal very similar structural regions called basal segments, lower stems, upper stems, and terminal loops. Based on these conserved structures, thermodynamic profiles of prime miRNA have been determined. The trochanter complex cleaves RNA molecule, squabble 2 helical turns away from the terminal loop and squabble 1 turn away from basal segments. In most analyzed molecules, this region contains unpaired nucleotides, and the free energy of the duplex is relatively high compared to lower and upper stem regions. Most pre-miRNAs don't have a perfect double-stranded RNA, dsRNA structure topped by a terminal loop. There are few possible explanations for such selectivity. One could be that dsRNA is no longer then TA21 base pairs activate interferon response and antiviral machinery in the cell. Another plausible explanation could be that the thermodynamic profile of pre-miRNA determines which strand will be incorporated into the dicer complex. Indeed, clear similarities between miRNAs encoded in respected 5' or 3' strands have been demonstrated. When dissect cleaves the miRNA stem loop, two complementary short RNA molecules are formed, but only one is integrated into the RISC complex. This strand is known as the guide strand and is selected by the Argonaute protein, the catalytically active RNAs in the RISC complex, on the basis of the stability of the 5' end. The remaining strand, known as the anti-guide or passenger strand, is degraded as a RISC complex substrate. After integration into the active RISC complex, mRNA's base pair with the complementary mRNA molecules and induce mRNA degradation by Argonaute proteins. The catalytically active members of the RISC complex. It is as yet unclear how the activated RISC complex locates the mRNA targets in the cell, though it has been shown that the process is not coupled to ongoing protein translation from the mRNA. Cellular functions. The function of mRNAs appears to be in gene regulation. For that purpose, a miRNA is complementary to a part of one or more messenger RNAs, mRNAs. Animal mRNAs are usually complementary to a site of the 3' UTR, whereas prime miRNAs are usually complementary to coding regions of mRNAs. The annealing of the miRNA to the mRNA then inhibits protein translation, but sometimes facilitates cleavage of the mRNA. This is thought to be the prime mode of action of plant mRNAs. In such cases, the formation of the double-stranded RNA through the binding of the miRNA triggers the degradation of the mRNA transcript through a process similar to RNA interference RNAi, though in other cases it is believed that the miRNA complex blocks the protein machinery or otherwise prevents protein translation without causing the mRNA to be degraded. miRNAs may also target methylation of genomic sites, which corresponds to targeted miRNAs function in association with a complement of proteins collectively termed the miRNP. This effect was first described for the worm C. elegans in 1993 by Victor Ambrose and co-workers. As of 2002, miRNAs have been confirmed in various plants and animals, including C. elegans, human, and the plant Arabidopsis thaliana. Work at the University of Louisville has resulted in the production of macroarrays containing all known, at the time of production, miRNAs for human, mouse, rat, dog, C. elegans, and Drosophila species, tools referred to as chips. Agilent has subsequently commercialized a human miRNA microarray. Genes have been found in bacteria that are similar to eukaryotic miRNA. Genes in the sense that they control miRNA abundance or translation by binding an mRNA by base pairing. However, they are not generally considered to be miRNAs because the DICE enzyme is not involved. In plants, especially RNA species, termed short interfering RNAs, siRNAs, are used to prevent the transcription of viral RNA. While this siRNA is double-stranded, the mechanism seems to be closely related to that of miRNA, especially taking the happen trans structures into account. siRNAs are also used to regulate cellular agents, genes, as miRNAs do. The gene activation, ts-RNA, can also activate gene expressions, a mechanism that has been termed small RNA-induced gene activation of RNAs, RNA-A. ds-RNAs targeting the gene promoters can induce potent transcriptional activation of associated genes. This was demonstrated in human cells using synthetic ds-RNAs termed small activating RNAs, saRNAs, but has also been demonstrated by endogenous microRNA. Detecting and manipulating miRNA signaling. The activity of an miRNA can be experimentally blocked using a locked nucleic acid oligo, a morpholino oligo, or a 2' O-methyl RNA oligo. Steps in the maturation of miRNAs can be blocked by steric blocking oligos. The target site of an miRNA or an miRNA can be blocked by a steric blocking oligo. miRNA and disease. Just as mRNA is involved in the normal function of eukaryotic cells, so has this regulation of mRNA been associated with a disease. Disease association in turn has led to increased funding, opportunities for academic research, and financial incentives for development and commercialization of mRNA-based diagnostics and therapeutics. After early commercialization aimed at academic research, support was established, and the initial research focus based on products and services requested was cancer and neuroscience research. During 2007, interests indicated by the products and services requested broadened to include cardiac research, virology, cell biology, and general and plant biology. miRNA and cancer. Several miRNA has been found to have links with some types of cancer. A study of mice altered to produce excess CMYC, a protein implicated in several cancers, shows that miRNA has effect on the development of cancer. Mice that were engineered to produce a surplus of types of miRNA found in lymphoma cells developed the disease within 50 days and died 2 weeks later. In contrast, mice without the surplus miRNA lived for over 100 days. Another study found that two types of mRNA inhibited the E2F1 protein, which regulates cell proliferation. miRNA appears to bind to messenger RNA before it can be translated to proteins that switch on and off. By measuring activity among 217 genes encoding miRNA, patterns of gene activity that can distinguish types of cancer cells can be discerned. miRNA signatures may enable classification of cancer. This will allow doctors to determine the original tissue type which spawned a cancer and to be able to target a treatment course based on the original tissue type miRNA. Yes. Profiling has already been able to determine whether patients with chronic lymphocytic leukemia had slow-growing or aggressive forms of the cancer. In 2008, the companies Rajan and Hexicon were working to commercialize this potential for miRNAs to act as cancer biomarkers. miRNA and heart disease. The global role of miRNA function in the heart has been addressed by conditionally inhibiting miRNA maturation in the murine heart and has revealed that miRNAs play an essential role during its development. miRNA expression profiling studies demonstrate that the expression levels of specific mitochondrial miRNAs change in diseased human hearts, pointing to their involvement in cardiomyopathies. Furthermore, studies on specific miRNAs in animal models have identified miRNA profiling has already been able to determine whether patients with chronic lymphocytic leukemia and slow-growing or aggressive forms of the cancer. In 2008, the companies Rajan and Hexicon were working to commercialize this potential for miRNAs to act as cancer workers. miRNA and heart disease. The global role of miRNA function in the heart has been addressed by conditionally inhibiting miRNA maturation in the murine heart and has revealed that miRNAs play an essential role during its development. miRNA's expression profiling studies demonstrate that expression levels of specific miRNAs in animal models have identified distinct roles of miRNAs both during heart development and under pathological conditions, including the regulation of key factors important for cardiogenesis, the hypertrophic growth response, the cardiac conductance. In 2008, academic work on the relationship between miRNA and heart disease had advanced sufficiently to lead to the establishment of a company, Rajan, with a primary focus on cardiovascular health and disease. So next would be section I1, we're dealing with the stability of proteins. The unique three-dimensional structure of each polypeptide is determined by its amino acid sequence. Interactions between the amino acid side chains guide the folding of the polypeptide to form a compact structure. 4 types of interactions cooperate in stabilizing the tertiary structures of globular proteins. Disulfide bonds. A disulfide bond is a covalent linkage from the sulfihydryl group SH of each of 2 cysteine residues to produce a cysteine residue. The 2 cysteines may be separated from each other by many amino acids in the primary sequence of a polypeptide, or may even be located on 2 different polypeptide chains. The folding of the polypeptide chains brings the cysteine residues into proximity and permits covalent bonding of the side chains. A disulfide bond contributes to the stability of the 3-dimensional shape of the protein molecule. For example, many disulfide bonds are found in proteins such as immunoglobulins that are secreted by cells. Note, these strong covalent bonds help stabilize the structure of proteins and prevent them from becoming denatured in the extracellular environment. Hydrophobic interactions. Amino acids with nonpolar side chains tend to be located in the interior of the polypeptide molecule, where they associate with other hydrophobic amino acids. In contrast, amino acids with polar or charged side chains tend to be located on the surface of the molecule in contact with the polar solvent. It's to be noted that proteins located in nonpolar lipid environments such as membrane, except with the reverse arrangement, that is hydrophilic amino acid side chains, are located in the interior of the polypeptide, whereas hydrophobic amino acids are located on the surface of the molecule in contact with the nonpolar environments. In each case, the segregation of R groups occurs that is energetically most favorable. 3. Hydrogen bonds. Amino acid side chains containing oxygen and nitrogen bound oxygen, such as in the alcohol groups of serine and 309, can form hydrogen bonds with electron-rich atoms, such as the oxygen of a carboxyl group or carbonyl group of a peptide bond. Formation of hydrogen bonds between molar groups on the surface of proteins and the aqueous solvent enhances the solubility of the protein. 4. Ionic interactions. Negatively charged groups, such as the carboxyl group COO in the side chain of aspartate or glutamate, can interact with the positively charged groups, such as the amino group NH3+ in the side chain of lysine. Protein folding. Interactions with between the side chains of amino acids determine how a long polypeptide chain folds in the intricate 3-dimensional shape of the functional protein. Protein folding, which occurs within the cell in seconds to minutes, employs a shortcut through the maze of all folding possibilities. As the peptide folds, its amino acid chains are attracted and repulsed according to their chemical properties. For example, positively and negatively charged side chains attract each other. Conversely, similarly charged side chains repel each other. In addition, interactions involving hydrogen bonds, hydrophobic interaction, the disulfide bonds all seek to exert an influence on the folding process. This process of trial and error tests many, but not all possible configurations, seeking a compromise in which attractions outweigh repulsions. This results in a correctly folded protein with a low energy. The role of chaperones in protein folding. It is generally accepted that the information needed for correct protein folding is contained in the primary structure of the polypeptide. Given that premise, it is difficult to explain why most proteins, when denatured, do not resume their native conformations under favorable environmental conditions. One answer to this problem is that a protein begins to fold in stages during its synthesis rather than waiting for synthesis of the entire chain to be totally completed. This limits competing folding configurations made available by longer stretches of nascent peptide. Addition, a specialized group of proteins named chaperones are required for the proper folding of many species of proteins. The chaperones, also known as heat shock proteins, interact with the polypeptide at various stages during the folding process. Some chaperones are important in keeping the protein unfolded until its synthesis is finished, or act as catalysts by increasing the rates of the final stage of the folding process. Others protect proteins as they fold so that their vulnerable exposed regions do not become tangled in unproductive encounters. Quaternary structure of protein. Many proteins consist of a single polypeptide chain and are defined as monomeri proteins. There's a difference in the alphabet. It looks like it's Finnish or some other language. Monomeri proteins, let's pronounce it that way. However, others may consist of two or more polypeptide chains that may be structurally identical or totally unrelated. The arrangement of these polypeptide subunits is called the quaternary structure of the protein. Note, if there are two subunits, the protein is called dimeric. If there are three subjects, trimeric, and several units, multimeric. Subunits are held together by non-covalent interactions, for example, hydrogen bonds, ionic bonds, and hydrogen interactions. Subunits may either function independently of each other or may work cooperatively as in hemoglobin, in which the binding of oxygen to one subunit of the tetramer increases the affinity of the other subunits for oxygen. Denaturation of protein. Protein denaturation results in the unfolding and disorganization of the protein's secondary and tertiary structures, which are not accompanied by hydrolysis of peptide bonds. Denaturing agents include heat, organic solvents, mechanical mixing, strong acids or bases, detergents, and ions of heavy metals such as lead and mercury. Denaturation may, under ideal conditions, be reversible, in which case the protein refolds into its original native structure when the denaturing agent is removed. However, most proteins, once denatured, remain permanently disordered. Denatured proteins are often insoluble and therefore precipitate from solution. And the next section would be I2, which is the stability of nucleic acids. In the Watson and Crick structure, the two chains or strands of the helix are antiparallel. The 5' 3' phosphodiester bonds run in opposite directions. Later work with the DNA polymerases provided experimental evidence confirmed by X-ray crystallography that the strands are indeed antiparallel. To account for the periodicities observed in the X-ray diffraction pattern, Watson and Crick used molecular models to show that the vertically stacked bases inside the double helix would be 0.34 nanometer apart, and that secondary repeat distance of about 3.4 nanometer could be accounted for by the presence of 10.5 nucleotide residues in each complete turn of the double helix. The two antiparallel polynucleotide chains of double helical DNA are not identical in either base sequence or composition. Instead, they are complementary to each other. Wherever adenine appears in one chain, thymine is found in the other. Similarly, wherever guanine is found in one chain, cytosine is found in the other. The DNA double helix or duplex is held together by two sets of forces as described earlier. Hydrogen bonding between complementary base pairs and base stacking interactions. The specificity that maintains a given base sequence in each DNA strand is contributed entirely by the hydrogen bonding between base pairs. The base stacking interactions, which are largely non-specific with respect to the density of the stacked bases, make the major contribution to the stability of the double helix. To the next section, J1, metabolism of carbohydrate, citric acid cycle. The citric acid cycle, Krebs cycle or tricarboxylic acid, TCA cycle, is the most important metabolic pathway for the energy supply to the body. About 65 to 70% of the ATP is synthesized in Krebs cycle. Citric acid cycle essentially involves the oxidation of acyl-CoA to CO2 and H2O. This cycle utilizes about 2/3 of total oxygen consumed by the body. The name TCA cycle is used since at the outset of the cycle, tricarboxylic acid citrate, cis-aconitate, and isocitrate participate. So that's why tricyclic. TCA cycle, the central metabolic pathway. The citric acid cycle is the final common oxidative pathway for carbohydrates, fats, and amino acids. This cycle not only supplies energy, but also provides many intermediates required for the synthesis of amino acids, glucose, heme, etc. Krebs cycle is the most important central pathway, connecting almost all individual metabolic pathways, either directly or indirectly. And the TCA cycle, the central metabolic pathway. Citric acid cycle is the most final common oxidative pathway for carbohydrates, fats, and amino acids. Repeat, the citric acid cycle is the final common oxidative pathway for carbohydrates, fats, and amino acids. This cycle not only supplies energy but also provides many intermediates required for the synthesis of amino acids, glucose and heme, etc. Krebs cycle is the most important central pathway connecting almost all the individual metabolic pathways, either directly or indirectly. A brief history of that. The citric acid cycle was proposed by Hans Adolf Krebs in 1937, based on the studies of oxygen consumption in pigeon breast muscle. The cycle is named in his honor, and he was awarded the Nobel Prize for Physiology and Medicine in 1953. Hans Adolf Krebs. Location of TCA cycle. The enzymes of TCA cycle are located where? In the mitochondrial matrix, in close proximity to the electron transport chain. So what happens? This enables the synthesis of ATP by oxidative phosphorylation without any hindrance. So that's how it's executed. Now, TCA cycle, an overview. Krebs cycle basically involves the combination of a 2-carbon acetyl-CoA with a 4-carbon oxaloacetate to produce a 6-carbon tricarboxylic acid citrate. In the reactions that ensue, the 2 carbons are oxidized to carbon dioxide and oxaloacetate is regenerated and recycled. Oxaloacetate is considered to play a catalytic role in citric acid cycle. Note this figure and take a screenshot, please. TCA cycle and open cycle. Krebs cycle is a cyclic process. However, it should not be viewed as a closed R circle, since many compounds enter the cycle and leave. TCA cycle is comparable to a heavy traffic circle in a national highway with many connecting roads, and each intermediate of this connecting cycle of the cycle connecting another pathway is a road. Reactions of citric acid cycle. Oxidative decarboxylation of pyruvate to acetyl-CoA by pyruvate dehydrogenase complex is discussed above. This step is a connecting link between glycolysis and TCA cycle. Some authors, however, describe the conversions of pyruvate to acetyl-CoA along with citric acid cycle. Pyruvate to acetyl-CoA along with citric acid. The events of TCA cycle are described here. From step 1, formation of citrate. Krebs cycle for proper starts with the condensation of acetyl-CoA and oxaloacetate catalyzed by the enzyme citrate synthase. This is an aldol condensation reaction leading to the formation of an intermediate acetyl-CoA, which hydrolyzes yields citrate. Citrate is freely permeable across the mitochondrial membrane. It serves as a good source of cytosolic acetyl-CoA, which is used for the synthesis of fatty acids. Citrate inhibits phosphofructokinase and activates acetyl-CoA carboxylase. The latter is the key enzyme in the fatty acid synthesis. Step 2 and 3, citrate is isomerized to isocitrate by the enzyme aconitase. This is achieved in a 2-stage reaction of dehydration, followed by hydration through the formation of an intermediate cis-aconitate. Step 4 and 5. Formation of alpha-ketoglutarate, the enzyme isocitrate dehydrogenase ICD, catalyzes the conversion oxidative decarboxylation of isocitrate to oxalosuccinate and then to alpha-ketoglutarate. The formation of NADH and the liberation of CO2 occurs at this stage. 3 types of ICD are known. 1. NAD specific, located only in mitochondria. 2. NADP specific, present in the mitochondria and cytosol. 3. The mitochondrial NAD plus dependent ICD is responsible for the conversion of isocitrate to alpha-ketoglutarate. Step 6. Conversion of alpha-ketoglutarate to succinyl-CoA. This occurs through the oxidative decarboxylation. It's catalyzed by alpha-ketoglutarate dehydrogenase complex. This enzyme is dependent on 5 cofactors: TPP, lipamide, NAD+, FAD, and CoA. The mechanism of the reaction is analogous to the conversion of pyruvate to acetyl-CoA. At this stage of the TCA cycle, second NADH is produced and the second CO2 is liberated. Step 7. Formation of succinate. Succinyl CoA is converted to succinate by succinate thiokinase. This reaction is coupled with the phosphorylation of a GDP to GTP. This is a substrate level phosphorylation. GTP is converted to ATP by the enzyme nucleoside diphosphate kinase. GTP plus ADP gives ATP plus GDP. So step 8, conversion of succinate to fumarate. The succinate is oxidized by succinate dehydrogenase to fumarate. This reaction results in the production of ADH2 and not NADH. This is due to the fact that the reducing power of succinate is not adequate to reduce NAD+, hence FAD is utilized. Formation of malate, the enzyme fumarase catalyzes the conversion of fumarate to malate with the addition of water, H2O. Step 10 would be the conversion of malate to oxaloacetate. Malate is then oxidized to oxaloacetate by malate dehydrogenase. The third and final synthesis of NADH occurs at this stage. The oxaloacetate is regenerated, which can combine with another molecule of acetyl-CoA and continue the cycle. Summary of TCA cycle. The events of Krebs cycle may be summarized as acetyl-CoA plus 3 NAD plus FAD plus GDP plus pi plus 2 H2O giving 2 CO2 plus 3 NADH plus 3H plus FADH2 plus GTP plus COA. Remember these formulas required by oxygen, requirement of oxygen by TCA cycle. There is no direct participation of oxygen in Krebs cycle. However, the cycle operates under aerobic conditions. This is due to the fact that NAD plus and FAD from NADH and FADH2 respectively required for the operation of the cycle can be regenerated in the respiratory chain only in the presence of O2. Therefore, citric acid cycle is strictly aerobic in contrast to glycolysis, which operates in both aerobic and anaerobic conditions. Energetics of citric acid cycle. During the process of oxidation of acetyl-CoA via citric acid cycle, 4 reducing equivalents, 3 as NADH and 1 as FADH2 are produced. Oxidation of 1 NADH by electron transport chain coupled with oxidative phosphorylation results in the synthesis of 3 ATP, whereas FADH2 leads to the formation of 2 ATP. Besides, there is 1 substrate level phosphorylation. A total of 12 ATP are produced from 1 acetyl-CoA. The inhibitors of Krebs cycle. The important enzymes of TCA cycle inhibited by the respective inhibitors are listed here. Enzyme: aconitase, alpha-ketoglutarate dehydrogenase, succinate dehydrogenase. Inhibitor: fluoroacetate, non-competitive, arsenide, non-competitive, malonate. Right. The most important synthetic anabolic reactions connected to TCA cycle are given here. Oxaloacetate and alpha-ketoglutarate respectively serve as precursors for the synthesis of aspartate and glutamate, which in turn are required for the synthesis of other non-essential amino acids, purines and pyrimidines. Succinyl-CoA is reused for the synthesis of porphyrins and heme. Mitochondrial citrate is transported to the cytosol where it is cleaved to provide acetyl-CoA for the biosynthesis of fatty acids, sterols, etc. Energetics of glucose oxidation. When a molecule of glucose 6-carbon undergoes glycolysis, 2 molecules of two molecules of pyruvate or lactate carbon are produced. Pyruvate is oxidatively decarboxylated to acetyl-CoA, 2-carbon, which enters the citric acid cycle and gets completely oxidized to CO2 and H2O. Just a second. Now, before that, we need to complete another part of the topic. Is a regulation of citric acid cycle. The regulation demands of ATP are crucial in controlling the rate of citric acid cycle. The regulation is brought about by either enzymes or the levels of ADP. 3 enzymes, namely citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate Dehydrogenase regulates citric acid cycle. Citrate synthetase is inhibited by ATP, and isocitrate dehydrogenase is activated by ADP and inhibited by ATP and NADH. Alpha-ketoglutarate dehydrogenase is inhibited by succinyl-CoA and NADH. Availability of ADP is very important for the citric acid cycle. This is due to the fact that unless sufficient levels of ADP are available, oxidation coupled with phosphorylation of ADP to ATP of NADH and FADH2 through electron transport chain stops, the accumulation of NADH and FADH2 will lead to inhibition of the enzymes as stated above, and also limits the supply of NAD plus and FAD, which is essential for TCA cycle to proceed. Amphibolic nature of the citric acid cycle. The citric acid cycle is the final common oxidative pathway in the living cells. The cycle also provides various intermediates for the synthesis of many compounds needed by the body. Krebs cycle is both catabolic and anabolic in nature and is regarded as amphibolic. Right now, the glucose plus 6O2 plus 3,8 ADP plus 3,8 PI gives 6 CO2 and 6 water plus 38 ATP, which means when a molecule of glucose is burnt in a calorimeter, 2780 kilojoules of heat is liberated. In the living system, energy is trapped, leading to the synthesis of 38 ATP, which is equivalent to 1159 kJ. 1 ATP has high energy bond equivalent to 30.5 kJ. That is about 48% of the energy in glucose combustion is captured for ATP generation. And next topic is under the section J2. It's called gluconeogenesis. The synthesis of glucose or glycogen from non-carbohydrate compounds is known as gluconeogenesis. Okay, synthesis of glucose or glycogen from non-carbohydrate compounds is known as gluconeogenesis. The major substrates or precursors for gluconeogenesis are lactate, pyruvate, glucogenic amino acids, propionate, and glycerol. Location of gluconeogenesis occurs mainly in the cytosol, although some precursors are produced in the mitochondria. Gluconeogenesis mostly takes place in the liver and to some extent in kidney matrix, about 1/10 of liver capacity. Importance of gluconeogenesis. Glucose occupies a key position in the metabolism, and its continuous supply is absolutely essential to the body for a variety of functions. Brain and central nervous system, erythrocytes, testes, and kidney medulla are dependent on glucose for continuous supply of energy. Human brain alone requires about 120 grams of glucose per day out of about 160 grams needed by the entire body. Glucose is the only source that supplies energy to the skeletal muscle under anaerobic conditions. All of the carbohydrates are synthesized from glucose, example lactose, amino sugars, etc. Glucose is converted to glycerol and utilized for the synthesis of fat. In fasting even more than a day, gluconeogenesis must occur to meet the basic requirements of the body for glucose and to maintain the intermediates of citric acid cycle. This is essential for the survival of humans and other animals. Certain metabolites produced in the tissues accumulate in the blood, example lactate, glycerol, propionate, example gluconeogenesis effectively clears them from the blood. Reactions of gluconeogenesis. Gluconeogenesis closely resembles a reversed pathway of glycolysis. Although it is not the complete reversal of glycolysis, essentially 3 out of 10 reactions of glycolysis are irreversible. The 7 reactions are common for both glycolysis and gluconeogenesis. The 3 irreversible steps of glycolysis are catalyzed by the enzymes, namely hexokinase, phosphorufractokinase, and pyruvate kinase. These 3 stages bypassed by alternate enzymes specific to gluconeogenesis are discussed now. Now, the conversion of pyruvate to pyruvate. This takes place in 2 steps. Okay, pyruvate carboxylase is a biotin-dependent mitochondrial enzyme that converts pyruvate to oxaloacetate in presence of ATP and CO2. This enzyme regulates gluconeogenesis and requires acetyl-CoA for its activity. Oxaloacetate is synthesized in the mitochondrial matrix. It has to be transported to the cytosol to be used in gluconeogenesis, whereas the rest of the pathway occurs Due to membrane impermeability, oxaloacetate cannot diffuse out of the mitochondria. It is converted to malate and then transported to the cytosol. Within the cytosol, oxaloacetate is regenerated. The reversible conversion of oxaloacetate and malate is catalyzed by malate dehydrogenase, an enzyme present in the mitochondria and cytosol. In the cytosol, phosphoenol pyruvate carboxykinase converts oxaloacetate to phosphoenol pyruvate. GDP or ITP, not ATP, is used in this reaction, and the carbon dioxide fixed carboxylase is liberated. For the conversion of pyruvate to phosphoenol pyruvate, 2 ATP equivalents are utilized. This is in contrast to only 1 ATP that is liberated in glycolysis for this reaction. Conversion of fructose 1,6 biphosphate to fructose 6-phosphate. The phosphoenol pyruvate undergoes the reversal of glycolysis until fructose 1,6 biphosphate is produced. The enzyme fructose 1,6 biphosphate phosphatase converts fructose 1,6 biphosphate to fructose 6-phosphate. This enzyme requires Mg2 ions. Fructose 1,6 biphosphatase is absent in smooth muscle and heart muscle. This enzyme is also regulatory in gluconeogenesis. Conversion of glucose 6-phosphate to glucose. Glucose 6-phosphatase catalyzes the conversion of glucose 6-phosphate to glucose. The presence or absence of this enzyme in a tissue determines whether the tissue is capable of contributing glucose to the blood or not. It is mostly present in liver and kidney, but absent in muscle, brain, and adipose tissue. The overall summary of gluconeogenesis for the conversion of pyruvate to glucose is shown below. Here 2 pyruvate plus 4 ATP plus 2 GTP plus 2 NADH plus 2 hydrogen plus 2 6H2O gives glucose plus 2 NAD plus 4 ADP plus 2 GDP plus 6 PI plus 6 hydrogen. After the formation of glucose, its conversion to glycogen occurs through the formation of uridine diphosphate glucose, UDP glucose, by the enzyme glycogen synthetase. Gluconeogenesis from amino acids. The carbon skeleton of glucogenic amino acids, all except leucine and lysine, results in the formation of pyruvate or the intermediates of citric acid cycle, which ultimately result in the synthesis of glucose. Gluconeogenesis from glycerol is liberated mostly in the adipose tissue by the hydrolysis of fats, triacylglycerols, the enzyme glycerokinase found in liver and kidney, absent in adipose tissue, activates glycerol to glycerol 3-phosphate. The latter is converted to dihydroxyacetone phosphate by glycerol 3-phosphate dehydrogenase, dihydroxyacetone. I repeat, dihydroxyacetone phosphate is an intermediate in glycolysis, which can conveniently be used for glucose production. Gluconeogenesis from propionate. Oxidation of odd-chain fatty acids and the breakdown of some amino acids, methionine, isoleucine, yields a 3-carbon propional-CoA. Propional-CoA carboxylase acts on this in presence of ATP and biotin and converts to methyl-malonyl-CoA, which is then converted to succinyl-CoA in the cycle involving the cycle involves synthesis of glucose in liver from the skeletal muscle lactate and reuse of glucose that's synthesized by the muscle of energy purposes known as KORAH cycle. In presence of B2 coenzyme, succinyl-CoA formed from propional-CoA enters gluconeogenesis via citric acid cycle. Propionate is an important precursor for gluconeogenesis in ruminant animals, such as cattle. Propionate is activated in propional-CoA, which is then converted to glucose as described above. Gluconeogenesis from lactate, KORAH cycle. The KORAH cycle. Lactate produced by active skeletal muscle is a major precursor for gluconeogenesis. Under anaerobic conditions, pyruvate is reduced to lactate by lactate dehydrogenase, LDH. Pyruvate, NADH plus H plus in the presence of LDH gives lactate and NAD plus. Lactate is a dead end in glycolysis. Since it must be reconverted to pyruvate for its further metabolism, The very purpose of lactate production is to regenerate NADH so that glycolysis proceeds uninterrupted in skeletal muscles. Lactate or pyruvate produced in the muscle cannot be utilized for the synthesis of glucose due to the absence of the key enzymes of gluconeogenesis, glucose 6-phosphatase and fructose 1,6-biphosphatase. The plasma membrane is freely permeable to lactate. Lactate is carried from the skeletal muscle through blood and handed over to liver, where it is oxidized to pyruvate. Pyruvate is so produced, is converted to glucose by gluconeogenesis, which is then transported to the skeletal muscle. Glucose-alanine cycle. There is a continuous transport of amino acids from muscle to liver, which predominantly occurs during starvation. Alanine dominates among the amino acids. It is postulated that pyruvate in skeletal muscle undergoes transamination to produce alanine. Alanine is transported to liver and used for gluconeogenesis. This cycle is referred to as glucose-alanine cycle. Regulation of gluconeogenesis, the hormone glucagon and the availability of substrates mainly regulate gluconeogenesis, as discussed here. Influenza glucagon. This is a hormone secreted by cells of the pancreatic islets. Glucagon stimulates gluconeogenesis by two mechanisms. Right. 2. Availability of substrates. Among the various substrates, glucogenic amino acids have stimulating influence on gluconeogenesis. This is particularly important in a condition like diabetes mellitus, decreased insulin level, where amino acids are mobilized from muscle protein for the purpose of gluconeogenesis. Now 3. Acetyl-CoA promotes gluconeogenesis. During starvation due to excessive lipolysis in adipose tissue, acetyl-CoA accumulates in the liver. Acetyl-CoA allosterically activates pyruvate carboxylase, resulting in enhanced glucose production. Gluconeogenesis from fat. It is often stated that glucose cannot be synthesized from fat. In a sense, it is certainly true, since the fatty acids, most of them being even chain, on oxidation produce acetyl-CoA, which cannot be converted to pyruvate. Further, the 2 carbons of acetyl-CoA disappear as 2 moles of CO2 in TCA cycle. Therefore, even chain fatty acids cannot serve as precursors for glucose formation. However, the glycerol released from lipolysis and the propionate obtained from the oxidation of odd chain fatty acids are good substrates for gluconeogenesis, as discussed here. Next section is J3, that's glycogen metabolism. Right? Now, the glycogen is the storage form of glucose in many animals. As starch in plants, it is stored mostly in liver, 6 to 8%, and muscles, 1 to 2%. Due to more muscle mass, the quantity of glycogen in muscle, 250 grams, is about 3 times higher than in the liver, 75 grams. The functions of glycogen. The prime function of liver glycogen is to maintain the blood glucose levels, particularly between meals. Liver glycogen stores increase in a well-fed state, which are depleted during fasting. Muscle glycogen serves as a fuel reserve for the supply of ATP during muscle contraction. Structure of glycogen in brief. Glycogen is a homopolysaccharide composed of alpha-D-glucose. Up to 100,000 residues may be present. The glucose units are held together by alpha-1,4 linkages. After 8 to 10 residues of glucose, a branch is formed with alpha-1,6 glycosidic linkage. Glycogen is stored as granules in the cytosol, where most of the enzyme of glycogen synthesis and breakdown are present. Glycogenesis. The synthesis of glycogen from glucose is glycogenesis. Glycogenesis takes place in the cytosol and requires ATP and UTP besides glucose. Synthesis of UDP glucose. The enzymes, hexokinase in muscle and glucokinase in liver, convert glucose to glucose 6-phosphate. Glucophosphoglucomutase catalyzes the conversion of glucose 6-phosphate to glucose 1-phosphate. Uridine diphosphate glucose, UDPG, synthesized from glucose 1-phosphate, and UTP by UDP glucose. Pyrophosphorylase, pyrophosphate PPI, produced in this reaction is hydrolyzed to inorganic phosphate, PI, by pyrophosphatase. This will ensure that the optimal synthesis of UDPG. Requirement of primer to initiate glycogenesis. A small fragment of pre-existing glycogen must act as a primer to initiate glycogen synthesis, it is necessarily found that in the absence of glycogen primer, a specific protein named glycogenin can accept glucose from UDPG. The hydroxyl group of the amino acid tyrosine of glycogenin is the site at which the initial glucose unit is attached. The enzyme glycogen initiator synthase transfers the first molecule of glucose to glycogenin. Then glycogenin itself takes up a few glucose residues to form a fragment of primer which serves as an acceptor for the rest of the glucose molecules. 3. Glycogen synthesis by glycogen synthase. Glycogen synthesis by glycogen synthase. Glycogen synthase is responsible for the formation of 1,4-glycosidic linkages. This enzyme transfers the glucose from UDP glucose to the non-residue-reducing end of glycogen to form alpha-1,4 linkages. The UDP release can be converted back to UTP by nucleoside diphosphate kinase. 4 would be the formation of branches in glycogen. Glycogen synthase can catalyze the synthesis of linear unbranched molecules with 1,4-alpha-glycosidic linkages. Glycogen, however, is a branched tree-like structure. The formation of branches is brought about by the action of a branching enzyme, namely glucosyl alpha-4,6 transferase amylo alpha-1,4 to 1,6-transglucosidase. This enzyme transfers a small fragment of 5 to 8 glucose residues from the non-reducing end of glycogen chain by breaking alpha-1,4 linkages to another glucose residue where it is linked by alpha-1,6 bond. This leads to the formation of a new non-reducing end besides the existing one. Glycogen is further elongated and branched, respectively, by the enzymes glycogen synthase and glucosyl 4,6 transferase. And now the formation of branches is brought about by the action of branching enzyme, namely glucosyl alpha-4,6 transferase amylo alpha-1,4 to 1,6-transglucosidase. This enzyme transfers a small fragment of 5 to 8 glucose residues from the non-reducing end of glycogen chain by breaking alpha-1,4 linkages to another glucose residue where it is linked by alpha-1,6 bond. This leads to the formation of a new non-reducing end besides the existing one. Glycogen is further elongated and branched respectively by the enzymes glycogen synthase and glucosyl 4,6 transferase. The overall reaction of the glycogen synthesis for the addition of each glucose residue is glucose N plus glucose plus 2 ATP gives glucose N plus 1 plus 2 ADP plus BI. Of the 2 ATP utilized, one is required for the phosphorylation of glucose while the other is needed for conversion of UDP to UTP. Glycogenolysis. The degradation of stored glycogen in liver and muscle constitutes glycogenolysis. The pathway for the synthesis and degradation of glycogen are not reversible. An independent set of enzymes present in the cytosol carry out glycogenolysis. Glycogen is degraded by breaking alpha-1,4 and alpha-1,6 glycosidic bonds. Action of glycogen phosphorylase. Now repeat, action of glycogen phosphorylase. The alpha-1,4 glycosidic bonds from the non-reducing ends are cleaved sequentially by the enzymic glycogen phosphorylase to yield glucose 1-phosphate. This process, called phosphorylasis, continues until 4 glucose residue remains on either side of branching point alpha-1,6 glycosidic